Welcome back, everybody. In this video lecture, we're going to look at genetic mapping in eukaryotes. And in the future, we'll look at genetic mapping in prokaryotes. To be successful in this endeavor, all you have to do is keep in mind what you've learned in the past, combine that with a tiny bit of extra understanding and a sprinkling of mathematics. And you're well on your way. So let's get started by going to the next slide. The next slide should be on the next page. There it is. So the agenda is quite straightforward. We're going to talk about what is linkage and define that. And then we'll move on to combine our understanding of linkage with what we already know about meiosis. And then we'll spend some time looking at genetic mapping. And we'll finish off by looking at mitotic recombination. Okay, so let's go to the next slide. And now let's look at what linkage is. So the best way to explain linkage would be to use an analogy. So the word link means a chain with links in it. So that chain is a single entity. So we can look at that piece of string as an entity, and if it has a handkerchief tied to one point in the rope, and it has another handkerchief of a different color tied to a different part of the rope, then wherever that rope goes, the two handkerchiefs follow. So the two handkerchiefs are linked together physically on the same piece of rope. And the only way to destroy that pattern would be to cut the rope between the two handkerchiefs, and then you would have no longer any linkage. So we take this analogy of a piece of rope, and we assume that that rope is a chromosome, and the two handkerchiefs are genes along that chromosome, then you're getting to the place where you need to be to understand the analogy of the rope to the chromosome. So we have a chromosome, and on that chromosome, we have a gene locus for green, and then we have a gene locus for red. Then the two genes, because they're physically on the same piece of DNA, are said to be linked on the same chromosome. So keep this in mind as we go to the next slide. But before we do, I want to mention something about the 7 traits which Mendel studied. So as you know from previous knowledge, Mendel studied 7 different characteristics. And he was pretty lucky that those 7 characteristics were genes located either on separate chromosomes, so there was no linkage, or, and here's the important part, that the 2 genes that he was studying were on the same chromosome, but at a great distance apart. So there's something here, there's a clue here as to what we need to look out for. So the distance between 2 genes is important in linkage, because during meiosis, something strange happens with the chromosomes. They get cut and pasted with partner chromosomes. So we call that crossover or crossing over. So suppose in Mendel's case, he was looking at the P color, and the gene for that was located way over there. And he was looking at the height of the plant, which was located on the same chromosome, but at the far end, such that the distance between them was great enough that they acted as though they were not linked. And that's the important thing out of today's lesson, right? If 2 things are close together, they tend to stay together on the same chromosome. If 2 genes are further apart, then there's a probability that in some of the offspring, they will be disconnected, unlinked. And if they're really far apart on the ends of the chromosomes, then they act as though they were on different chromosomes, just like if they were on 2 separate chromosomes. So this is a very, very important concept that you need to take forward in order to understand anything else. Okay. So Mendel was lucky, but he was such a genius that maybe he would have worked it out I think. So let's move on and talk about meiosis. We should know by now that crossing over is a phenomenon of zygotine, where the homologous chromosomes during prophase 1 are brought together and enzymes are released into the cell to promote the process of crossing over. In my opinion, the best way to understand this is to break it down into parts. Let's just start with one chromosome. One chromosome. Let's make that chromosome a particular color. Let's make it red. So here it is. So here's your chromosome, and it contains a centromere somewhere in the middle. Now, in a diploid organism, we have a homologous chromosome. So we need to put the second chromosome in. Normally, we keep the two chromosomes a similar or same color. But today, we're going to change the color because it's going to be important. So let's make the partner chromosome a slightly different color. Let's make it purple. So there it is, right? And it will also have a centromere at the same location, and it'll have genes along its length. So let's just deal with one gene, and let's call that gene big A, and it sits right there on that locus. Now suppose the homologous chromosome contains an alternative allele of that gene. So it's going to be small a in our example. So that's what the normal diploid cell looks like. Then it begins to undergo meiosis. So in meiosis 1, let's put that down next. So in meiosis 1, the chromosomes, after they've been duplicated, will come together. So let's draw the duplicated chromosomes, so that makes sense. So here we are. Here's one chromosome, and it's been duplicated, like that. And it's connected still at the centromere. And then we have a second chromosome. Let's draw the second chromosome as well, and this time in purple. And this chromosome, of course, will be brought to sit side by side with its homologue during leptotene, the phase before zygotene. Okay, so we have that now sitting right here, like this. And let's put the genes on there so we keep track of them. So the red will contain big A, and the purple will contain small a at that particular locus. Okay, the locus is right there. Okay. With respect to the chromosomes are going to now be encouraged by the chemistry of the cell to exchange chromatids. The cell will prevent sister chromatids from exchanging between themselves because that would be just a waste of time. But it wants to encourage the exchange of chromatids between the homologous pair. So let's put that down in a different color, right? So let's use a blue color to indicate a region of crossover. So suppose the crossover is down here. So the inner two chromatids have switched DNA. So what happens subsequently during anaphase 1? So let's write that down. So anaphase 1, the homologous chromosomes will be pulled apart. And the red chromosome will go one way, like this. And now you will see that it carries a part of the purple chromosome right there because of the crossover, right? And then the other chromosome, the one on the right, will be pulled to the right in this particular instance. Let's draw that in. And then after a certain point there, it'll be carrying the exchanged DNA molecule on its chromatid. So genetically, that would now be this. So right here, we would have big A. Right there, we'd have small a. Right here, we'll have big A. Right there, we'll have small a. And then, of course, that will then segregate into the two daughter cells. So let's put those cells in right now. So here they are. Here's one cell. Here's the other cell. So this one goes into this one, and that one goes into that one. And then we'll have a situation where this process will continue and then go through meiosis 2. And in meiosis 2, the individual sister chromatids will be then converted to chromosomes and placed inside each of those daughter cells. Okay, so what we'll end up at the end is something like this. So let's take the red one, put that one in here like this, and a bit of red in there. And then we have a whole bunch of red in here with a bit of purple. So I'm just putting these chromosomes inside that cell. So let's continue to do that and finish it off. So we have this, and then we have that. Okay, a bit more purple right here. And then what's going to happen next is that these chromosomes will end up inside the respective haploid cells. So this one will contain mostly purple with a bit of red, the top one. This one will contain entirely purple, and then this one here will contain entirely red, and this one here will contain mostly purple. Mostly red with a bit of purple. There you go. Okay, and that's how we explain a single factor, a single characteristic, a single gene with two alleles segregating into the haploid cells, right? In this case, the gametes. All right, so now that we understand that, let's look at two genes at the same time. So I'm just going to add to this figure here because we can, and I'll use a different color. I'll use blue to indicate that. So here's blue. Let's say we have a second gene. Let's put it up here. And in this case, let's call it big B on the red chromosome and small b on the purple chromosome. Now it's very important to remember where these chromosomes came from. Okay, so the red one, this one here, it came from, let's say, the mother. Okay, so that's your maternal chromosome. The other chromosome, the purple one, that came from the father. So that's your paternal chromosome. So that's going to be very important because we have this term now to understand. Parental, right? So if you look at it, the mother gave big A, big B. That's the combination of alleles that it passed on into its segregation into this individual, right? So we're looking at the F1 here. This is the baby, and the baby is undergoing meiosis to produce its gametes, right? So the parents of that baby, the P1, or just the P generation would be right here. So those are the parents. So this baby inherited big A, big B from its mother, small a, small b from its father. So those are the parental combinations of chromosomes. If you now continue to put the B chromosomes, the B alleles on the chromosomes, you'll find that we have big B here and big B there, and small b there and small b there. Crossing over, in this case, will shuffle the order of those genes. Or will it? So we said that crossing over was down here. And if that's still down there, then in this case, we will still have big B on here, and we'll have small b there. We'll have big B there, and we'll have small b there. So when the gametes are produced, the one at the top, this one here, the one I'm highlighting, that gamete will pick up the combination of big B, big A, because they're sitting right there. And the one below it will be small a, small b. And then the one below that will be the reciprocal. That will be A, B, and the bottom one will be A, B. A, B, capital, A, B, lowercase, right? So in this case, it's going to be capitals. These are lowercase, that's lowercase, and these are capitals. So with respect to those two genes, nothing has changed. They still look parental. Even though there's been a crossover, it hasn't exchanged lowercase with uppercase. Uppercase stays with uppercase, A and B, lowercase stays with lowercase. So we call these parental type chromosomes. So as far as A and B is concerned, they still have the same combination as the parents. So there's nothing odd there at the moment. But suppose the crossover was between A and B. And that can happen because it's a mostly random process, right? So instead of the crossover being down here, suppose the crossover was somewhere between A and B. What would happen then? So let's quickly draw that. And we won't draw everything. We just need to draw the middle part. So we get rid of the far side because we don't need it anymore. So that just confuses us. And then we just follow the chromosomes, right? So we're going to assume that there's a crossover right there as indicated between, this is the one crossover that we're looking at, right? So the crossing over system came along in zygotine and it put a crossover event right there. So what's going to happen now? So let's draw that. So we're going to ignore the crossover at the bottom and just assume that the crossover above has taken place. So let's draw this very, very rapidly. All right. So what we'll have is we'll have the outside chromatid unchanged. Then we'll have the inside chromatid carrying near the tip a piece of red. Okay. Oh, wrong word, a piece of purple. So now I wish I'd chosen alternative colors. Then we have the right-hand side chromatid. That's all going to be all purple. And then the one, its sister chromatid will be purple up to that point, and then it would be red because it did the crossover at this place, right? So that's a crossover. So now let's look at the alleles on the chromatids. So let's do that in black. So here we still have big A, and here we still have big A. Over there we have small a, likewise there. Now let's do the top. Here we have big B. Now here we don't have big B. Because of the crossover, we now have small b. And over here we have big B, and over there we have small b. Now when these chromosomes are separated by the end of the first meiosis, and then they enter second meiosis and then get segregated into the gametes, what will end up is this. So let's draw that very quickly, because this is the crux of everything, right? So this chromosome there will be this one here. Okay, there's your centromere, there's the rest of the chromosome. Then the next chromosome there will be this one here. So it'll be mostly red with a bit of purple on top. Okay, let's put the purple right there. And then we have the, let's put the inside one next. So we have the inside one, which is mostly purple with a bit of red on top, right? And we'll put the red in the second. And then we have the final one down here, which will be all purple with the centromere. And let's put the red in place so we don't get that lost. There you go. Now let's put the alleles in place, okay? So for the top chromosome, we have right there, we have big A, and right there, we have big B. So this is this one here. Then for the bottom chromosome right there, let's do the same thing. So right there, we have small a, right there, we have big B. Okay, now let's go for this red one here. Now we see that we have big A, and now we have small b. Reshuffling. And likewise, for this particular chromosome here, we'll have small a there and big B there. So this chromosome will be big A, big B. This chromosome will be big A, small b. This chromosome here will be small a, big B, and the bottom one will be small a, small b. So this still looks like the parental chromosome that was inherited by the F1. This still looks like the parental chromosome that was inherited by the F1. and is passing it unchanged onto the next generation. But here's the crux of the whole chapter. Now we have a new combination. We have a new combination of big A with small b, which didn't exist when the chromosomes came in to this individual. So those are called recombinant. Those are called recombinant chromosomes because they've been recombined into a new pattern that didn't exist prior. And that's the crux of everything to do with mapping, genetic mapping, as we're learning in this chapter, right? So we can measure, believe it or not, we can measure how many gametes of each type are produced by looking at the offspring, the F2. So by measuring the F2 and mathematically applying an understanding to that, we can calculate how often this crossover event occurs, and then extend that understanding to mapping the genes, how far apart the genes are on the chromosomes. That's the ultimate goal of this particular video lecture, to get a value for the distance between the A locus and the B locus. And we can do that by applying genetic mapping strategies, right? So what we have at the end is we have something called genetic recombination. It's nothing more than crossing over that leads to a shuffling of chromosome segments. So take a pause and compare this diagram that I've drawn on this slide to what would happen if the two genes, A and B, were on different chromosomes. What would happen then? Would you get the same outcome? That some gametes will be big A, big B, some will be big A, small b, some gametes will be small a, big B, and then the fourth class of gamete will be small a, small b. Isn't that what we have seen so far? And in the past, we told you that you get 25% of each category. So 25% of the sperm or the eggs will be of one type, 25% of the second, 25% third, and the final 25%, the fourth type. So that's what Mendel got when he did his experiments. And that led him to make the conclusions that he did. But if those two genes, A and B, are close together on this one chromosome, then they're going to exhibit linkage, like two handkerchiefs on a rope. Where A goes, B should follow, unless there's a crossover to separate them. That's the premise of linkage. So pause the video if you need to digest that knowledge, to digest that information. If you want to go back to a two-factor cross and see what the outcome was, this will be a good time to pause this video. So I'm going to move on, and then we'll look at some other areas of understanding that we need. Let's begin by explaining things very methodically. Let's begin with some parents. And the first parent is going to be homozygous dominant for A and homozygous dominant for B. And we're going to mate this parent with another parent that's going to be homozygous recessive for A and homozygous recessive for B. Then the gametes that are produced by the first parent will be big A, big B. And the gametes produced by the second parent will be small a, small b. And then in the F1, these two will come together to give us a double heterozygote. So this. Okay. Now let's breed it to itself. Let's self that, cross it to itself, and then we'll end up with a Punnett square. And in the Punnett square, we will end up with how many classes? 4 classes of phenotype. If big A is dominant and big B is dominant, you have it. So 9 to 3 to 3 to 1 from the 16 box Punnett square, right? So those 9 will be big A with something, big B with something. And then the 3 will be big A with something with small b, small b. And then the other 3 will be small a with big B and something. And then the final one will be the double homozygous recessive. So there we go. So this is familiar. And we're assuming that the 2 genes are not linked. They're on separate chromosomes or they're far apart. And then we will get certain outcomes. But what would happen if the genes were linked? How would that change the outcome? And that's the key. So what we compare what we know to what's going to happen in this particular setting. So let's look at that. Suppose we produce 1,600 F2s. 1,600. How many of those would be of each of these classes, these phenotypic classes? So we would say that the expected number, based on our understanding of no linkage, would be 900 of those type, 300 of those type of phenotype, 300 of that, and then 100 of that. And the total should add up to 1,600. So those are the expected. But if the genes were on the same chromosome and they were reasonably close to each other, we could get a different outcome. So we could say that the observed outcome, we got something such as the following. Suppose we got 1,200 of these, 100 of these, 100 of these, and the remaining, we have 200 of these. So that adds up to 1,600. So that departure from the expected, the observed, would indicate something odd going on. And the most likely thing in this particular example is linkage. That gene A and gene B are physically on the same chromosome, close enough that there's crossing over taking place and generating some variation, but not enough. There's not enough crossover to unlink the big A with, say, the big B in the example that we gave earlier. So this is how you discover the distance apart of the 2 genes by doing the mathematics. Let's go to the cytology again, because it will help us understand this. Let's draw some chromosomes, right? So we're going to draw 2 chromosomes. And again, in natural conditions, there'll be homologous pairs, but in this case, we're just going to keep it very simple. So here's one chromosome, and let's draw the other one, this time in a green. So when we started this exercise, we said the 3 genes were on the same chromosome. That's what the data suggests, because the observed and the expected are not the same, right? So that tells us we're dealing with linked genes. So the question then becomes, in which order are the genes on the chromosome? So they could be one of 3 different orders. So the first order they could be maybe there's an R here, S, and W. That's one order, right? Or maybe there's S, R, W. Or maybe the W is in the middle. So there's S, W, R. So those are the 3 orders, right? So in this case, W could be on this side or on that side. It doesn't matter. All we want to know is relatively what's in the middle. So those are the 3 possibilities. So the data will help us determine this. Remember what we told you earlier, right? The largest class was R++ at 359. And then the reciprocal of that was plus SW at 351. Those were the most common types of progeny. So this tells us that the chromosomes must have had that orientation. So let's put that in place. So let's get rid of this. I have to draw that chromosome again, sorry. This doesn't take long, there it is, okay? So what we're saying is this, based on this data here, we know that the R was with 2 pluses on one of the chromosomes in the parent. And then the reciprocal of that was S plus W. Now we don't know if R is in the middle yet or not. We don't know that. But this is one possibility. But we do know that the whole situation started with the red chromosome carrying the 2 wild types and the R mutant allele, and the green chromosome carrying the reciprocal. They had 1 wild type and 2 mutant alleles. And then crossing over took place. There, and again, multiple times in between. And that crossing over would shuffle the genes. So by looking at the smallest class and the largest class, we can determine which order the genes are, the correct order of the genes. That's what we're interested in first. The secret sauce is to work out how do you get the small r away from the 2 pluses. So what we're looking for is how do you make this? And that would be reflected by the lowest class. Remember, we had 6 of those. And then we had R, S, W at 4. So the lowest classes will allow you to predict who's in the middle. Now, the only way you can get from here to there is if R is in the middle. If R is in the middle, because it takes double crossover to get that. So you can see that right here, right? So if this is 1 chromosome, then it does this and back again. That's the only way you can get R to come down and join the small s and the small w. And the reciprocal of that would be this. This up here and then back again, right? So that'll give you small s, small r, and small w on the same chromosome. And then, of course, the reciprocal of that will be giving you plus, plus, and plus. So the smallest number allows you to work out who's in the middle, and the largest numbers tell you which combination or the parental chromosomes. So this data, half the data is revealing something. But that's not all. We can go further. We can now work out the map distance by using the other values, the other 4 values, as we did on the previous slide. So we came up with 20% and 10%, right? So which is 20%, which is 10%? Well, we worked out that we worked out so far, let me change the pencil. We worked out so far that R is in the middle, and then S is on one side and W is on the other side of the chromosome. And we also worked out from the previous slide that the recombination frequency between R and W was 10%, and the recombination frequency between R and S was 20%. So if we were to redraw the figure at the top in a very quick fashion, this one here, we could now redraw things. We don't know which side the centromere is on, by the way, so we're going to just draw it without the centromere, all right? So we could say something like this. Here's one chromosome, and here's the other chromosome. And we can now say that W lies over here, the W gene. Then 10 units away, we have the R gene. And then 20 units away, we have the S gene. Like I said, the centromere could be on this side or that side. We don't know that until we do a further analysis. So this 3-factor cross has allowed us to calculate 2 important things. The order of the 3 genes, who's in the middle, and their distance apart. Now you can confirm this by doing a further set of mathematics, but that's just a confirmation step. To make sure that what you're looking at is real, the scientists can then apply the chi-squared analysis to confirm that the expected data and the observed data are really different, and there is linkage going on. But if the numbers, the expected numbers, are very close to the observed numbers, then the chi-squared analysis may reveal that's just a random variation. But if you apply chi-squared to this, you will find out for sure that this is real. There's something going on, and that something is linkage. Linkage because 3 genes are on the same chromosome, and they're close together. And the numbers that we gave you, the 20% and the 10%, these numbers here, 20% and 10%, they can be translated into map units. So a 1% recombination frequency is equivalent to 1 map unit, or 1 centimorgan apart on the chromosome. So chromosomes don't add up to 100% because the length of the chromosomes can vary. But for small distances, we can use these map distances to relate the relative distance of genes close together. Then if you wanted to find out what relationship W had with T and V, you could do another set of genetic crosses, and that may reveal that those 2 genes are on the same chromosome if there's linkage. And if they're close together, then there will be some kind of linkage data suggesting a disparity in the observed data compared to the expected data. So as you can see here, we've calculated the map units. We've also calculated the order of the genes. Practice makes perfect, so you have to do about 3 to 5 problems like this and understand what each step is doing, and then you should be able to conquer these principles. What I would need to do is quickly finish off. So you can also get recombination, believe it or not, in mitosis. It doesn't normally happen, but cells can make mistakes. So normally what happens is chromosomes line up at the metaphase plate without the homologs sharing any information. But every now and again, the homologs may line up like that accidentally for a time, and then they can exchange chromatids. And that exchange of chromatids between homologous chromosomes can lead to recombination. And if that happens in the tissue of the body, then you get mosaicism, right? So you get hybrid tissue. And that is seen in examples that we had to explain in the past. So twin spotting, where tissue on the body of a Drosophila has a weird characteristic. It has a difference in genotype between 2 genes, or phenotype, I should say, called twin spotting. That was the first sign that mitotic recombination was a force to consider in rare instances. So once again, the main gist of this lecture was to understand genetic linkage and how linkage can then interfere with the expected number of progeny when we're looking at multiple genes. Thank you so much.